Proteins are the component in the cell that exert the function. And the proteins, they work not by themselves, but they work in complex with other proteins. So the MolBoolean method that we developed is a method to visualize the amount of a certain protein and if it interacts with another protein. So you get information both on free protein and complex bound proteins. MolBoolean, if you compare to methods like in Situ that measures the amount of complex protein complexes, MolBoolean also gives information about how much free protein you have in a cell. There's other methods like a FRET where you can measure both free and complex bound protein, but MolBoolean in this case has a signal amplification, so you can detect single molecules. MolBoolean has advantage that you can look in single cells. You can see amount of protein and protein complex in a cell. So that is a tool that you can study signal transduction, for example. If you have free protein and then you have the signal from outside, it's a relay of protein-protein interactions that goes down to the nucleus, for example, where you induce expression of target genes. So it's better understand basic functions in the cell. So precision medicine, it means that we all respond differently to drugs. I mean, it depends on our genotype and how a drug will affect us. But of course, depending on that, you have that all the proteins also will differ a little bit. So it might be that one protein differs from another protein from another person. So having MolBoolean, we can look at molecular events in a single cell, allows us to develop new drugs that can be more targeted for certain individuals. I think it can be applicable for almost anything, but of course cancer will be one example when you have a change in the protein conformation due to a mutation, how that affects signal transduction. Other examples is virus infecting a cell, how does it affect the protein machinery in the cell? So I think it can be used to see this molecular effect of a drug. So if you have a drug that can interfere with the protein-protein interaction, MolBoolean will see, like, do you affect the amount of protein or do you affect the protein-protein interaction?